At EHA, we'll be presenting the phase 3 ENHANCE trial. So this is a 500 patient trial with the primary endpoints of both CR rates and overall survival in an all-comer high-risk MDS group that's intermediate or higher. And this was agnostic to P53 mutation status. I think, unfortunately, as everybody's aware, the study did press release negative, but I think key is, we've worked very diligently, both the investigators and the company, to present these data as soon as possible. The question is, what are the big differences between our phase 1 experience, where response rates appear to be double, versus historical and quite durable, CRs and median overall survival, again, both in mutant and P53 groups. So what happened? I think part of the challenge is the toxicity. I think the on-target anemia was underappreciated early in the trial. There was a major change where we did implement sort of this day 1, day 4 hemoglobin threshold of greater than 9 and repeat checks afterwards. That did seem to mitigate some of that, but again, the trial had already accrued significantly, and a substantial number of patients came off of therapy early. And if you can't keep people on sort of optimal combination, at least through 6 cycles, the Azacitidine control arm has won now numerous times from that perspective. So I think that was an underappreciated, toxicity. I think, potentially were there other imbalances. The trial did not stratify based on P53 mutation status, which I think may have been a flaw of the design as well. And then it sort of imbalances to transplant. So if a lot more patients get to transplant in one arm than another, that can clearly change survival quite dramatically, and that was really also the case. So I think the bottom line is, clearly the trial was not positive. Magrolimab does not, have a role or future sort of in the frontline treatment of higher risk, MDS or AML patients. But I'm hoping that from all of this, we've learned. And so again, there's at least 2 other agents in randomized phase 2 trials. And again, we need to look at some of these questions differently. At least all the other agents do not have this sort of on-target anemia. And again, can we keep people on combination therapy long enough to truly see the synergy of these agents? And I'm hoping, again, we can still move the field forward with targeting CD47 or CD47 combination strategies.